Welcome to video series on protein metabolism. This video is on detoxification of ammonia and formation of urea along with urea. Once the protein performs its function, they undergo degradation. Now, the degradation time varies for different proteins. For example, the lifespan of ornithine decarboxylase enzyme associated with the polyamine metabolism is only 11 minutes when compared to insulin, which has a lifespan of few hours. And this, when compared to hemoglobin, has got a lifespan of 120 days. And whereas the lengths of proteins they are present during the entire lifetime of the individuals. So once the protein completes its functions, they undergo degradation. Now the degradation differs from extracellular and intracellular proteins. Extracellular protein are taken up by endocytosis. They enter into the lysosome. In the lysosome, they are acted upon by cathepsin, which are 18 in number, represented from A to T, and these cathepsin are activate pH, acidic pH of 5. The proteins are converted to individual amino acids, and this does not require energy. So this mechanism is also known as ATP independent protein degradation. Later on, these amino acids are released into general circulation and they are taken up by different target tissues for protein biosynthesis to form the biologically important proteins. Now the intracellular proteins, they are tagged with another protein molecule called ubiquitin. They enter into a complex called proteasome complex, and once the intercellular protein enters into this complex, ubiquitin is released back. Now they are acted upon by various enzymes, and these enzymes require hydrolysis of ATP to break down this protein to individual amino acids. So this is known as ATP dependent ubiquitin mediated intracellular protein. Now, coming to formation of ammonia, what are the sources of ammonia in our body? So, we have two important sources, major source and the minor source. Major source comes from catabolism of amino acid, where there occurs removal of amino group from all the different amino acid by transamination process, and this is followed by oxidative deamination releasing ammonia. So, the minor sources for ammonia include the catabolism of purine bases, adenine, guanine, catabolism of pyrimidine bases, cytosine, uracil, thymine, and catabolism of amino sugars, and synthesis by the intestinal bacteria. So this ammonia is also detoxified to urea and liver, and this urea gets excreted by our renal system. Coming to catabolism of amino acid, which is the major source of ammonia, how is this taking place inside our body? Now, in all the peripheral tissues and organs, transamination reaction is taking place. So, transamination is a reaction where amino acid gets converted to keto acid. For example, alanine gets converted to its keto acid pyruvate. Alanine donates the amino group to alpha ketoglutarate, which is a keto acid, and after accepting the amino group, alpha ketoglutarate gets converted to glutamate, which is the new amino acid or the non-essential amino acid. Now, this transamination is taking place in all the cells, all the tissues and all the organs. Now, this glutamate is transported to liver. So, glutamate is a major transportation form of amino group from peripheral tissues to liver. Whereas in brain, the amino group is carried by glutamine. So, glutamine from the brain reaches liver. So, here it is acted upon by glutaminase enzyme, and glutamine gets converted to glutamate. Now, so this glutamate undergoes oxidative deamination in liver because this is catalyzed by glutamate dehydrogenase enzyme, and this enzyme is present only in the hepatocytes and not in any other cells. Now, so glutamate gets oxidized to alpha ketoglutarate releasing ammonia, and simultaneously NAD is getting reduced to NADH, and this ammonia gets detoxified to urea, and urea is excreted by our kidney. So this is about the catabolism of amino acids, and this is the major source of ammonia in the body. Now, so this ammonia is detoxified by three different lines of defense mechanism. So the first line of defense is trapping of ammonia by glutamate in all the tissues and glutamine in brain. So the second line of defense is removal of the ammonia from glutamate by oxidative deamination and from glutamine by glutaminase enzyme. And the third and final line of defense is formation of urea. Let us try to understand how is this urea formed in the body. Now this is urea cycle. Now this is it is called as bicycle because part of the cycle is operating in mitochondria and part part of the urea cycle is operating in cytoplasm. The first step in urea cycle is condensation of ammonia with carbon dioxide to form carbamyl phosphate. This is catalyzed by enzyme carbamyl phosphate synthetase 1, and this is the rate limiting step of urea cycle. Now this requires energy in the form of hydrolysis of 2 ATP to form 2 ADP. Now in the next step, carbamyl phosphate reacts with ornithine catalyzed by the enzyme ornithine transcarbamylase to form citrulline. Now this citrulline comes out of the mitochondria into the cytoplasm through a transport protein which is specific for citrulline. Now this citrulline in turn reacts with aspartate to form argininosuccinate catalyzed by the enzyme argininosuccinate synthetase. Now this also requires energy of hydrolysis of ATP to form AMP. So this equals releasing pyrophosphate. This is equal to utilization of 2 ATP molecules. In the next step, argininosuccinate is acted upon by lyase enzyme to form arginine and fumarate. Now this arginine is acted upon by argininase enzyme forming ornithine and urea. Now ornithine is recycled back into the mitochondria using a specific transport protein. Now this here the fumarate, which is formed in the previous step through a special transport protein, it gets channelized back into the mitochondria. Here fumarate is acted upon by fumarase enzyme to form malate. Now malate get oxidized to oxaloacetate, and simultaneously NAD is getting reduced to NADH. Now this NADH get channelized to ETC producing 3 ATP molecule. Now this oxaloacetate undergoes transamination to form aspartate. Now through a specific transport protein, aspartate is released back into the cytoplasm so that it can combine with citrulline. Now, so the urea that is formed in the ammonia is released into the general circulation, and the concentration of blood urea is around 20 to 40 milligram per deciliter. Now this is taken up by the kidney. It is freely filtered. 40% of the urea gets reabsorbed and remaining gets excreted, and around 15 or 20 to 30 gram per day of urea gets excreted by the kidney. So the molecular formula for urea is CONH2 NH2. There are 2 nitrogen atoms, and the source of these 2 nitrogen atoms comes from ammonia and aspartate. Coming to energetics of urea cycle, the first rate limiting step catalyzed by carbamyl phosphatase synthetase requires hydrolysis of 2 ATP. Next, condensation of citrulline to aspartate to form arginosuccinate also requires ATP hydrolysis releasing inorganic pyrophosphate, and this also amounts to 2 ATP. So totally 4 ATPs are required for the formation of urea from ammonia. Now the oxidation of malate to oxaloacetate generates NADH, and this when channelized into ETC generates 3 ATP. So what is the net energy yield from urea cycle? So for the formation of urea from ammonia, 4 ATP are needed, and the oxidation of malate to oxaloacetate generates 3 ATP. So the net energy yield is 1 ATP. Now coming to regulation of urea cycle, the fine regulation is mainly done by N-acetyl glutamate, and this N-acetyl glutamate is formed by condensation of glutamate with acetyl CoA to form N-acetyl glutamate, and this act as a positive allosteric effector of carbamyl phosphatase synthetase 1. So the next type of regulation is by compartmentalization. This is because part of the urea cycle enzymes are present in mitochondria, and part of the urea cycle enzymes are present in cytoplasm. So this will bring about compartmentalization. This is because fumarase enzyme inhibits argininase. So this is prevented by placing fumarate and fumarase enzyme in mitochondria and argininase enzyme in cytoplasm. That is how compartmentalization brings about regulation of urea cycle, and these two are fine regulation. Coarse regulation is done by starvation. So whenever we are starving, all the urea cycle enzymes are stimulated. For example, starvation stimulates carbon phosphate synthetase 1, ornithine transcarbamylase, arginosuccinic acid synthetase, arginosuccinic acid lyase, argininase enzyme. All these are stimulated in condition of starvation. So these are the 3 mechanisms by which urea cycle is regulated. To disorders of urea cycle, they can be classified as congenital disorders and acquired hyperammonemia. Congenital disorders are usually caused by deficiency of the enzymes of the urea cycle. Now these include hyperammonia type 1. This is due to deficiency of carbamyl phosphate synthetase 1, which is the rate limiting enzyme of urea cycle. Now this disorder is characterized by very high levels of ammonia, and as ammonia is not getting detoxified to urea, now this inhibits the influx of release of glutamine from brain. So there is increased glutamine in brain and CSF, which impairs the cerebral functioning, and this leads to development of severe mental retardation among these children. So the next enzyme deficiency is deficiency of ornithine transcarbamylase, and this results in hyperammonia type 2. This is also characterized by very high levels of ammonia and glutamine, and one special characteristic feature of this hyperammonia type 2 syndrome is orotic aciduria. This is because when this ornithine transcarbamylase is deficient, now this carbamyl phosphate gets channelized to pyrimidine nucleotides, where carbamyl phosphate gets converted to carbamyl aspartate dihydroorotate. This gets converted to orotate, and this gets excreted in urine, causing orotic aciduria. So orotic aciduria is one of the characteristic feature of hyperammonia type 2. So the next disorder is citrullinemia due to deficiency of arginosuccinic acid synthetase enzyme. This also is characterized by increased ammonia and increased citrulline. So the first three enzyme deficiency are characterized by hyperammonia and urea, and these disorders, urea cycle disorders, the first three enzyme disorders are usually characterized by hyperammonia, encephalopathy and alkalosis. Now this hyperammonia is very dangerous because this depletes the alpha ketoglutarate, so this is not available for TCA cycle as substrate, and as we all know, TCA cycle is mainly concerned with the generation of reducing equivalents which are channelized to ETC to provide energy. So there is decreased energy metabolism in these individuals, and further there is increased glutamine because the influx of glutamine from brain is decreased. Now this glutamine is highly osmotically active substance, and it causes cerebral edema. Further, increased glutamine there decreases the glutamate formation. So decreased glutamate with the decreased formation of GABA inhibitory neurotransmitter severely alters the neuronal signaling in the brain, resulting in CNS dysfunction. So resulting also results ultimately leads to encephalopathy. So the other characteristic feature of urea cycle disorder is alkalosis. This is because increased ammonia directly stimulates the respiratory center, inhibits the respiratory center, resulting in respiratory alkalosis. Coming to the next enzyme deficiency, of arginosuccinate lyase, resulting in arginosuccinic aciduria. Apart from increased ammonia and increased accumulation of arginosuccinic acid, so this gets accumulated in the blood and excreted in urines, resulting in arginosuccinic aciduria. One more characteristic feature of this condition is trichorexis nodosa, characterized by friable and brittle hair. So the last enzyme deficiency results in hyperargininemia due to deficiency of arginинase. This is characterized by increased arginine in blood, CSF and urine. So these are the congenital disorders. Coming to the acquired hyperammonia, now this is mainly seen in liver failure or the hepatic coma. So the biochemical basis here is, normally what happens is the intestine generates urea by the intestinal bacterial metabolism. Now this ammonia goes to liver, where it gets detoxified to urea. But in liver cell failure, what happens is, now this ammonia is not taken up by the liver. Instead, it enters the general circulation, resulting in portal systemic shunting, and this ammonia results in all CNS dysfunctions. Coming to the clinical features of liver failure or the hepatic coma, first here the liver is damaged, and this results in jaundice because of increased release of bilirubin inside the circulation. resulting in yellowish discoloration of skin, sclera, and conjunctiva. Next is ascites because of decreased protein synthesis, hypoproteinemia, hypoalbuminemia. When the protein synthesis is decreased and when the levels are less than 5 gram per deciliter, or when the albumin is decreased less than 2 gram per deciliter, it results in accumulation of the albumin in the interstitial compartment, especially around the abdominal wall, manifesting as edema, and in the peripheral tissue in the legs it manifests as edema. So hemorrhage occurs because of decreased or defective synthesis of the clotting factors. Spider nevi is characteristic dilatation of the arterioles or the capillaries in the skin due to decreased clotting factors and decreased metabolism in the liver. Next is because of increased ammonia, there is CNS dysfunction, and this results in altered sensorium, seizures, and encephalopathy. So this was about acquired. Now that you have understood all about urea cycle, I am right now giving you one technique for long-term retention of your memory, which is known as SQ3R technique. S stands for survey, Q stands for question, followed by 3R means read, revise, and recall. So survey and question is here you are rapidly reading your textbook, which your textbook of biochemistry you are following. You have to give a very rapid reading instead of thorough reading. This is followed by question. You have to ask this question: why this topic is important from exam point of view and from patient point of view. This has already been covered in the video that is presented, so no need to so you are free from this task. So the next is you need to watch the video, then you have to just mentally revise the concept that was covered in the video, and this should be followed by a recall. So you can mentally recall key points from the video. Now to facilitate long-term retention, so one more concept I have introduced in this video is self-assessment, which will help you to evaluate how good you have understood the topic. So I have presented around 10 MCQs so that this will help you to understand how good you have understood the topic. That is one thing. Wherein what I will be doing is I'll read the stem of the MCQ, and I'll give you a couple of seconds for you to mentally think about the answer. So this will be followed by me explaining the answer. Go. So the major toxic effect of ammonia in our body is on. Please think. 3, 2, 1, and the answer is brain. So this is because glutamine accumulates in the brain and it will deplete the alpha ketoglutarate, which is needed for the TCA cycle. So no TCA cycle, no energy, and neuronal dysfunction. Apart from that, glutamine is osmotically active and it will cause swelling of the cerebral cells. That is how it exerts. Next question: the principal organ responsible for detoxification of ammonia is 3, 2, 1, and the answer is liver, because liver has got the enzyme glutamate dehydrogenase, which is not present in any other tissue. So only liver hepatocytes have got the capacity to convert or detoxify ammonia to urea. So next question: which amino acid acts as a non-toxic transport form of ammonia from brain to liver? 3, 2, 1, and the answer is glutamate. So next question: urea cycle occurs in cytosol only, mitochondria only, nucleus, both mitochondria and cytosol. 3, 2, 1, and the answer is both mitochondria and cytosol, because urea cycle enzymes are placed partly in the mitochondria and partly in cytoplasm. Question: the first and rate limiting enzyme of urea cycle is 3, 2, 1, and the answer is carbamyl phosphate synthetase 1, which will condense the ammonia and carbon dioxide to form carbamyl phosphate. Next question: which molecules acts as obligatory activator of carbamyl phosphatase synthetase 1? 3, 2, 1, and the answer is N-acetyl glutamate, which is formed from condensation of glutamate and acetyl-CoA. So this N-acetyl glutamate is a positive allosteric modifier of the key enzyme of urea cycle, that is carbamyl phosphatase synthetase 1. Next question: nitrogen atoms of urea is derived from 3, 2, 1, and the answer is both ammonia and aspartate. So ammonia and aspartate as there are two nitrogen atoms in the urea, because the molecular formula of urea is NH2CONH2. So first nitrogen atom is derived from ammonia and the other nitrogen atom is derived from aspartate. Next question, which urea cycle intermediate is formed in mitochondria and transported to cytosol? 3, 2, 1, and the answer is citrulline. Citrulline is mainly formed in the mitochondria, and there is a special transport protein for citrulline which transports citrulline from mitochondria to cytoplasm for further operation of the pathway. Next question, final step of urea cycle produces 3, 2, 1, and the answer is ornithine and urea. And ornithine in this urea cycle acts as a catalyst. It enters the cycle and comes out without any change, so it acts as the catalyst of urea cycle. Next question is, energy required for the formation of one molecule of urea is 3, 2, 1, and the answer is 4 ATP. 4 ATP is because the very first reaction, rate limiting step, catalyzed by carbamyl phosphatase synthetase 1, where ammonia combines with carbon dioxide to form carbamyl phosphate, requires 2 ATP. And the next step where 2 more ATPs are required is conversion of citrulline and aspartate to form arginosuccinic acid, catalyzed by the enzyme arginosuccinic acid synthetase. Next question, net energy from urea cycle is 3, 2, 1, and the answer is 1 ATP. So from the previous MCQ we know that there are 4 ATPs that are utilized in this reaction, and the fumarate that is formed in the urea cycle is transported to mitochondria, where fumarate gets converted to malate. Malate gets oxidized to oxaloacetate, generating NADH, and this NADH when channelized into ETC generates 3 ATP. So 4 minus 3 is equal to 1 ATP. So the net energy yield from urea cycle is 1 ATP. So thank you. This was all about urea cycle, its disorders, and the MCQs to make you understand the topics better. So the next video will be on glycine metabolism. So these are the most probable exam question asked. Describe the steps of the urea cycle with energetics, regulation, and associate disorders. Most probably it will be asked as a straightforward long essay question, and the question can also be asked as describe the detoxification of ammonia. So this was all about ammonia and urea cycle. Thank you. Please do like, share, and subscribe.